So, hi everyone. Thanks for joining us virtually. I'd like to thank Fatigatio, Angehörige for inviting me to speak for about half an hour here on some neuroscience issues in ME. I'm Michael VanElzakker. I'm a neuroscientist at Massachusetts General Hospital and Harvard Medical School. This is just me saying I have no disclosures, no financial interests here. So I wanted to talk a little bit about what's happening in the world, I think, which primarily is the COVID-19 pandemic. Obviously long COVID has brought a lot of attention into chronic illness, viral initiated chronic illness. We don't like to use the term post-viral because that has an assumption embedded into it. But in chronic illness that begins with an infection, it's really been all over the newspapers. And there was a study led by Hannah Davis and a patient cohort in which they just did a sort of a online tracking of what people experienced when they had long-term symptoms that didn't remit quickly. And the symptoms here, you can see here the percentage of people after 6 months. The symptoms really sort of circled, I think, if you will, around the feeling of being sick and failure of recovery from exercise, fatigue, autonomic symptoms and pain. So this sickness, autonomic symptoms, and pain. And for people that know about ME, that's probably somewhat familiar. I think those of us who knew about ME, kind of knew that SARS-CoV-2, the virus that causes the COVID-19 illness, was going to lead to these problems in a certain percentage of people. And, if you look at the symptoms of ME, this is the international consensus criteria, they really also center around sort of the feeling of being sick, fatigue, failure of exercise recovery, and autonomic problems and pain. So sickness, autonomic problems, pain, sickness, autonomic problems, pain. It seems to be sort of a trend here. And I won't go through the symptoms because you're all more familiar with them than I am. So, Dr. Proal and I of the Polybio Research Foundation wrote a review article about long COVID in which we went through some potential mechanisms by which this may be happening that we think are similar to ME. And it really goes back to the naming of myalgic encephalomyelitis, which was in the Lancet in 1956, where the author of this article argued that based on some preliminary findings in cerebral spinal fluid, some known and some unknown viruses, by the way, that they should call it benign myalgic encephalomyelitis. Not benign meaning that it wasn't harmful, but benign meaning that it wasn't tending to kill people like the polio illness was. Actually, Bjorn Sigurdsson argued in a letter responding to this article that maybe we shouldn't name it after neuroinflammation because that assumes a mechanism and we don't quite know if that's the mechanism. That was the MD that was studying the Akureyri outbreak in Iceland. But here's really where the name ME was coined. And I think it's important to note that this isn't really new. So a lot of new quote unquote pathogens, viruses, when they become known to people, we realize that they are associated with a long-term consequences, so long-term illness. So here's an example of Ebola, so-called post-Ebola syndrome, where survivors reported pain, headache, and ocular problems or eye problems. Post-Zika, including Guillain-Barré, post-MERS, which is another coronavirus. If you take a look, it's very similar to the symptoms, right? Reduced exercise capacity, some symptoms of anxiety and depression, but sort of failure to recover from exercise. Post-SARS syndrome, which is the SARS-CoV-1 virus, included musculoskeletal pain, fatigue. So these are sort of overlapping symptoms with ME, right? So we get a little bit into that here. Just so you know, it's sort of interesting that this post-Ebola, it turned out that at least in some cases, the virus simply persisted. The virus survived, even though it wasn't ripping through people's bodies like it does acutely. So here's an example. Remember how I mentioned that post-Ebola syndrome included problems with vision, the eye, ocular problems. So here's an American doctor that months prior had been declared free of Ebola because they did blood tests. Blood tests aren't very good at detecting pathogens, unfortunately. So he said at the time, he thought the problems were an immune-related Ebola complication, right? So this is what we hear all the time. Maybe the immune system just kind of went crazy. It was trying to fight the virus and the immune system went crazy and never settled down. Well, it turned out that he had live virus, including a virus that survives in the testes, which is somewhat privileged site that doesn't have quite as strong of an immune response. So, this idea of viral reservoirs, some amount of viruses surviving, might be important in a certain percentage of patients that have these long-term consequences. So here's an example where they talked about the SARS-CoV-2 virus persisting and being found in gut after COVID-19. So here's an example where they found the virus surviving in gut in some people. This is after acute COVID. Now, interestingly, we've found, Dr. John Chia has found enteroviruses in stomach biopsies in ME diagnosed patients who have long-term problems. So what are some of the mechanisms by which a virus might be causing long-term problems? This is an obvious one, right? That it simply just doesn't go away. That's an obvious clue. But another thing that happens that viruses do is that they immunosuppress. Now that seems sort of counterintuitive, right? You'll hear all the time that these problems are caused by the immune system going crazy, going up too much, right? It's too strong. But I would argue that that's not really accurate. What actually happens in these cases is that the immune system becomes inefficient because it's been sabotaged by an acute pathogen. So here's an example of the SARS-CoV-2 infection sabotaging the interferon type 1 cytokine system. Now, interferons are the primary antiviral cytokine. So one of the things that happens is viruses and bacteria and fungi that survive in humans often do so by sabotaging the immune system, allowing other pathogens to take advantage. So one thing that might happen with an acute, and by the way, other viruses do this too. One thing that might happen with an acute infection is that it stirs up a gut dysbiosis. So the organisms that live in everyone's gut may turn more pathogenic, more virulent. They can change their gene expression to express more virulence factors. These organisms are called pathobionts. So how can all these different problems, viral persistence, making other viruses survive and change, making other bacteria survive and change, how can they cause all these similar symptoms? Well, I would like to focus on an area of the brain called the dorsal brainstem. Dorsal, like the dorsal fin of a shark, right on the back. So here's a scan. This is actually my brain from the side. And I wanted to focus in on the brainstem, which is here. And if we enhance that and make it a little bit larger, this is what it looks like. Now, an interesting thing about this area of the brain is it's really filled with a lot of important anatomy. So here I've overlaid some anatomical structures onto the dorsal brainstem. Well, let's make it a little bit bigger so you can see even better. And what I want to do is to take you through a little tour of some of these structures and then explain why they might matter. There's a lot of structures and circuits that are in close proximity here. They're overlapping. And the fact that they're overlapping and may be affected by illness, inflammation, I think might matter for long-term consequences. So the nucleus of the solitary tract is an area of the brain where the vagus nerve enters. So the vagus nerve shares the same Latin root as the word vagabond, meaning to wander, right? So it wanders all over the body, all over the trunk, and it has what's called chemoreceptors. So in other words, it can detect chemicals, including chemicals that are associated with inflammation, like cytokines, which you may have heard of. What happens when it does that is that it sends a signal up to the brain that says, hey, there's infection, there's a problem in the body, go to bed, you're sick. You should save your energy because you should be fighting this, this infection or perhaps this injury. So a majority of the cell bodies of this vagus nerve are here in the nucleus of the solitary tract. Now look how that overlaps with all of these other structures. And that's going to make sense in a moment when I talk a little bit more about that. Right nearby is this area called the area postrema. Now, normally the brain has what's called a blood-brain barrier, which protects the brain from anything that might be just circulating throughout the body in blood. But there's little areas where there's a fenestration or window in the blood-brain barrier that allows larger molecules to be detected by the brain. And this is one of those areas, area postrema. It allows sampling of bigger molecules, macromolecules. It's really important for the feeling of nausea and for vomiting, which I know for some chronic illness patients is an ongoing issue. So here's the vagus nerve. Let me talk a little bit more about that. It's really important because if you take a rat, for example, and you inject a rat with, let's say, bacteria, like E. coli, or you inject a rat with something that mimics E. coli, like LPS or lipopolysaccharide, or you inject a rat with something that mimics the response to bacteria, like cytokines, the rat will act sick, right? The rat will not run around, it won't socialize, it won't eat as much unless you've cut the vagus nerve. And then the rat doesn't know that it's sick. Its brain doesn't know that its body is sick. It will still run on a wheel like it normally does. It'll eat the normal amount, et cetera, et cetera. So the vagus nerve is really important for transmitting the reality of being sick, which is detected in the body, up to the brain to tell the body, hey, go to bed, you're sick. Interestingly, too, by the way, this circuit is more sensitive in women than is in men, broadly speaking. So the afferent vagus nerve, which means sensory from body to brain, has a couple different functions that I think are really interesting. So it detects peripheral catecholamines. So what that means is hormones like adrenaline or epinephrine that are produced by your adrenal glands, right? Adrenal, right on top of your kidneys. Those don't easily cross the blood-brain barrier. So what actually happens is that those chemoreceptors of your vagus nerve detect the adrenaline or epinephrine, and that's what causes the so-called adrenaline response. Importantly, which I've already mentioned a little bit, is it detects local immune signaling molecules. What that means is if you have a little infection in your stomach or a little infection in your lungs or a little infection in your throat, the vagus nerve has so many branches that one of those branches is likely to be right next to that infection. And it will detect the inflammation, and then it will tell your brain, hey, the body is sick, go to bed, you're sick. So that's a really important function. It surveys the gut microbiome. Remember, I talked about how the fact that during an initial infection, sometimes the gut microbiome can sort of become dysbiotic. This is one of the things that the vagus nerve detects. And all of these things initiate what's called the central illness response, which is what I've alluded to before, which includes the feeling of being sick. It includes some dysautonomia, so changes in heart rate, for example, are common. And it also includes increased pain. Now, remember the three symptoms that I focused on before: sickness, dysautonomia, and pain. Those are, generally speaking, part of the initial acute immune response. So we think that that may be really important because these are all functions of the vagus nerve. And this is detectable with the technique PET. And I am running a PET study along with some other people across the United States are also running PET studies to measure this. So let me just explain a little bit how this works. So in the body, there are immune cells like white blood cells, such as monocytes and macrophages. They're on the other side of the blood-brain barrier from the brain. When they detect pathogens, whether that's herpes viruses or the SARS-CoV-2 virus or Borrelia or other bacteria, anything like that, they activate and they release pro-inflammatory cytokines. So these are those immune molecules that I was saying before are detected by the vagus nerve. Now, these are somewhat big protein molecules that don't easily just flow across the blood-brain barrier into the brain. So some of them can get in that way, but generally speaking, that's not how we think about them. They're not released in very, very large numbers that allow them to flow through the body like a hormone would. So instead of that, sometimes they can go right across that circumventricular organ, the place that has the little window in the blood-brain barrier, like area postrema, which is that area that I showed you in the dorsal brainstem. So interestingly, the area postrema can also just drag immune cells across. It's called active transport. So it actually brings macrophages and sometimes monocytes across into the brain to try and help fight infection is the idea. Now, interestingly, there's a German research group that found in the dorsal brainstem, right near where the vagus nerve enters, SARS-CoV-2 virus in COVID cases. Now, this is acute COVID, not long COVID, but it shows that, yes, these areas of the brain, these important areas of the brain, can become infected and probably drive severe symptoms. This is examples of where the virus was detected. The lead author of this study, I think did a very careful job. He went through a very difficult peer review process before this was published. So this is the story of the neurological infection with the SARS-CoV-2 virus. They think it largely affects the blood vessels, which might cause long-term problems. So let's go back to the neuroscience for a bit. So besides the cytokines going right across into the brain, either directly through the blood-brain barrier or around the blood-brain barrier in places like area postrema, cytokines are also detected by the vagus nerve. These are the chemoreceptors. So now again, because the vagus nerve wanders all over the body, it can detect a very small amount of cytokine in a local infection. It doesn't have to be circulating in the blood, which is part of the reason that cytokine studies in ME have been really inconsistent. It's actually not the smartest place to measure. You do your best. I mean, where else are you going to look, right? It's a difficult problem. But generally speaking, they are paracrine, not endocrine signaling factors. In other words, they don't flow through the blood to have their effect. They affect cells that are really local and nearby. When the vagus nerve detects those with its chemoreceptor, it actually triggers what's called a mirror response of glial activation on the brain side of the blood-brain barrier. Glia, which is Greek for glue, because it used to be thought that they just held neurons together. Glia are the immune cells of the central nervous system. There's a whole bunch of different types, but they activate when this signaling pathway is excited. And that's what drives the feeling of being sick. That's the sickness response that makes people want to stay in bed and feel a little extra soreness and pain, and sometimes even some autonomic problems. So just to summarize really quick, the vagus nerve's highly branched structure allows detection of local immune signaling. So in other words, the immune signaling happens really locally. It doesn't have to be all through the body in order for it to have a profound effect on how people feel. It actually doesn't have to be an infection. It can be injury too, which is pretty interesting and important perhaps. So it triggers a mirror response of glial activation that's measured by PET scanning. And here's an example of a study, a small study that a lot of groups are now trying to replicate by Nakatomi 2014. So back to the circuitry here. Near the nucleus of the solitary tract in the area postrema is this area called the dorsal motor nucleus. So this is a site of autonomic control. So it's where the parasympathetic nervous system projects out into the body. It's really important for controlling things like heart rate and things like that. So we know that autonomic problems are important in many chronic illness cases. The dorsal reticular nucleus, which is down here, again, overlapping with the nucleus of the solitary tract in area postrema, is really important for the transmission of inflammatory pain. Now, this is another thing that we keep talking about, is a lot of patients that have chronic illness have sort of diffuse pain all over their body. They feel sore, their muscles feel sore. It's possible that the problem is actually out, near the skin where the pain is sensed, but it's also possible that maybe this circuit in the brain is a little sensitized, and that's part of what's causing the enhanced feeling of soreness and pain. Here is the ventrolateral medulla. So this controls blood pressure and breathing. Again, these are autonomic problems that a lot of chronic illness patients have. And the nucleus ambiguus also controls heart rate. And again, you can see how these areas are all just really overlap. So how do we know that they all become activated when there's a sickness or an ongoing inflammatory response? Well, we know from rat studies. So Lisa Goller and Ron Gekema were at the University of Colorado when I was there, when I was just an undergraduate and a technician. They were on the faculty there, and they, along with Linda Watkins, particularly, really helped us understand how these brain circuits work, how the feeling of being sick is transmitted from the body responding to something, responding to almost anything really, to the brain, causing us to feel sick. So here's an example, just one example of a study in which they squirted bacteria down a mouse's throat to mimic food poisoning, right? So this is actually acute infection, right? So what they did here was to then take the mouse and slice up the brain and look to see where neurons were active. The black here is where neurons were active. Over on the left column is the mice that were infected with essentially food poisoning bacteria. And on the right was just saline. So you can see these areas are much more active when the mouse feels sick. And take a look at these areas. They're the exact areas that I was just talking about. The nucleus of the solitary tract, area postrema, the dorsal motor vagus, ventrolateral medulla, and reticular nucleus. So these are all areas that are really important in the feeling of being sick. This is what makes even a mouse, when there's some issue like an infection, feel sick. They don't want to socialize. They don't want to run. Their heart rate actually changes, and they just feel sick, right? Interestingly, here, again, the left side is the infection. The right side is saline. Left side infection, right side saline. These are areas that are involved in the feeling of stress and anxiety. So a lot of patients, not all, but a lot of patients feel stress and anxiety. Well, that can be part of being sick. It's literally activated by the same circuits in the brain. So here, for example, is the paraventricular nucleus of the hypothalamus. This is where stress hormones, this is the cascade that starts stress hormone response. The bed nucleus of the stria terminalis, the insular cortex, the amygdala. These are areas that are involved in the feelings of being stressed and anxious. So I think it's really important to remember that if patients are anxious, it doesn't mean that the anxiety is causing the sickness. A lot of times the sickness is causing the anxiety. Not all patients feel that, but a lot of them do. So just with this rat study, we've seen that it can cause sickness, autonomic problems, and pain, right, in those same exact circuits. And if there's long-term problems, then maybe it's due to long-term, problems in the body. So here's an example of how the vagus nerve detects gut dysbiosis. So in other words, if you've got your gut bacteria are out of balance, something came along and stirred them up, that is detected by the vagus nerve. That's what it does. And I talked about some of these issues in a paper years ago in which I talked about a specific infection, but it's the same mechanisms. It doesn't have to be a specific infection. It's really anything that causes an inflammatory response can drive these symptoms. Another interesting factor that I think is important in neuroscience is something called glial priming. Now, I've talked about glia before. So this is an example of microglia, which are what's called the resident macrophages of the brain. So macrophage means big eater. Those immune cells that gobble up bacteria and viruses and things like that. They're the ones that live in the brain. And they kind of look like this. And when they detect a pathogen, they activate. So what they do or importantly, I guess, also when they detect cytokines. And that could be cytokines that are triggered by that circuitry that I described through the vagus nerve, where the vagus detects something in the body, maybe some problem in the gut, and then it triggers a mirror response in the brain. The microglia here will detect that and then respond by activating. When they activate, they sort of draw in their long, skinny arms. They turn this sort of fatter, thicker sort of phenotype. That's sort of what they look like. And then importantly, they release a whole bunch of stuff, right? So one of the things they release are cytokines themselves, which is a form of immune signaling. So that's symbolized here by these little triangles that I've been using to symbolize cytokines. Another thing that they do is to release things that excite neurons. So things like glutamate, bradykinin, prostaglandins, ATP, nitric oxide, a whole bunch of things that you don't have to memorize, but they excite nearby neurons. And I think that that might be part of why patients with ongoing inflammatory issues have trouble concentrating. It's really important that the brain is really efficient and has a really good signal to noise precision. And this is one of the things that can really disrupt that. And importantly, it releases what's called the translocator protein. The translocator protein is just part of the immune response, but it's the thing that we measure with PET scanning. That's that type of brain scanning that we can do to tell us whether or not there's neuroinflammation happening in a patient. And that's, one of the, that is a study that I'm conducting on an ongoing basis. After this happens with glia, they enter a state called primed, where they're just like a little bit more sensitive. It doesn't have to last for the rest of someone's life, but glia, generally speaking, live pretty long. And they aren't really replaced by circulating cells that replace them. They tend to be replaced by replicating themselves. So you can see that if glia enter a state that's a little dysfunctional, that might last a long time. What primed means is that when a second challenge comes, maybe of the same exact level of that first challenge, right? The same amount of cytokines or the same amount of infection, a much larger response happens. So this may be part of why, for example, patients can end up feeling really sensitive. So a small amount of disruption can really make a patient feel really bad for a long time. They may be really sensitive to chemicals and things like that. I talked a little bit about this in a review article that we wrote a couple of years ago, my lab and I, the importance of measuring these things properly, studying them carefully. The methods are really difficult. It's very easy to miss important signal and things like that. So this is worth reading, I think, if you're interested in these topics. And with that, I just, want to thank some of my collaborators. So Ken Kwong, Phoebe Chan are helping me with a study of blood vessel responses in ME, so the brain's blood vessels, how they respond in people with the ME diagnosis versus controls. John Palamani and Roberta Scloco are both brainstem imaging experts that are helping me with a study, and Marco Loggia and Ava Ratai are both experts in studying neuroinflammation with brain scanning, and we're doing, again, a PET study, a PET MR study together that's been going on for a little while, and we're right in the middle of it. So with that, danke sehr, thank you very much, and I'll hopefully see you live in a little bit. Thank you.